Hi everybody, welcome to your bacteria video notes. So we're going to start by talking about some general characteristics of bacteria, then we'll get into the different kinds and also different diseases associated with them. So general characteristics. So you should know already that bacteria cells are prokaryotic, and the word prokaryotic means before nucleus. So these cells do not have a true nucleus. They do have genetic material inside, but they don't have a nucleus to contain it. And there are two domains of bacteria within which are two kingdoms. So domain Archaea has kingdom Archaea Bacteria, which are your extreme environment bacteria. And then domain Bacteria has kingdom Eubacteria, which are more of the normal bacteria or the ones that we're more familiar with. First, let's talk about Archaea Bacteria. So within this kingdom, there are three different types of extreme bacteria that exist. So there's thermoacidophiles, halophiles, and methanogens. So first, thermoacidophiles. When we think thermo, you should think like temperature, right? So kind of hot. So hot, and then acidic environments. So these ones live in places like sulfur hot springs, even thermal ocean floor vents that are really, really deep underwater. So these bacteria like this hot, acidic environment. Halophiles, on the other hand, like salty areas. So the Great Salt Lake, the Dead Sea, these have much higher salt concentrations than other saltwater bodies of water. But these bacteria are aerobic, so they do still like oxygen, they just prefer higher salt concentrations. And then lastly, methanogens. So these are the opposite of halophiles in that they prefer anaerobic conditions. So they do not really need oxygen to survive, and instead they give off this methane gas. And these are common in sewage treatment plants, swamps, bogs, kind of smelly locations is where you would typically find these. So now we have eubacteria, and a lot of what we're going to talk about is bacteria that belong to this kingdom. So these are the most numerous animals on the entire planet, and they're known as the true bacteria. They have these really strong cell walls made of something called peptidoglycan, which is kind of web-like, but it's still very strong and supportive for them structurally. Some of these bacteria do have cilia and flagella to help them move. So flagella is kind of like a tail and cilia are the hairs. And fun fact for you, these are actually the most studied organisms. They can produce 4 generations in under 20 minutes with optimal conditions. That's like you, your parents, all your aunts and uncles, your grandparents, and your great-grandparents can produce all of those generations in less than 20 minutes. And some examples here, I'm sure you've all heard of E. coli, Streptococcus, which causes strep throat, and then Lactobacillus, which is actually in some dairy products. Let's look first, before we get any further, at just kind of the general structure of a bacteria cell. And remember, this is typical, okay? It's typical bacteria cell. This is not necessarily every single bacteria cell out there. So the cell has 7 major structures that we're going to focus on that you're responsible for being able to label and locate. First, we have that flagellum. So flagellum is singular. Multiple would be a flagella, which is just the long tail. The pilus is single. Pili is plural. So every one of these little hair-like structures. There's your DNA or your genetic material inside. Ribosomes inside. If we remember, ribosomes make proteins. Those are important. And then lastly, we have the 3 different surrounding membranes of this cell. So we start with a plasma membrane or a cell membrane, which is your innermost membrane. That is followed, and it's white here. You could not read that, but it's white. And then you have a cell wall, which is dark pink. I'm just going to write my hand. And then lastly, the outermost structure is your capsule, which is your light pink structure. And that has these 3 different structures to surround it, protect it. So now that we know a little bit about the structure, let's talk about just some common shapes and arrangements of bacteria. And there are way more out there than we're going to learn in class, but let's just kind of focus on some of the majors. So we're going to start here with this picture. So we have a cocci, a bacilli, and a spirilla shape. So cocci is kind of like a sphere shaped, bacilli is a rod or an oval, and then spirilla is spiral. Within that, you can have different arrangements. So if you look over here, the cocci or the coccus bacteria can be mono, meaning one, diplo, paired. Staphylo is a clump, write that there. And then strepto is the other one we're going to focus on, which is a chain. You can also have some of these arrangements within bacillus or the rod-shaped bacteria. So once again, you see the diplo, you see the strepto, and then you have something called palisades, which is kind of like a chain, but they're lined up differently. And then there's different curve shapes of bacteria as well. We're just going to focus on the spirilla spiral. Now that we know what they look like, let's talk about how we get more, right? So bacteria can reproduce two ways. They can reproduce asexually or sexually. Binary fission is the term we use for asexual reproduction in bacteria. And this is just kind of sort of like mitosis. So the cell's going to copy its genetic material, it's going to grow larger in size, and then it's just going to split in half. And this is honestly the most common form of prokaryotic reproduction. Most bacteria are going to reproduce this way. And you see the examples here, E. coli and staph, they use this method. And if we look all the way over here, that's just a time lapse of those bacteria expanding and growing. So you can see how they kind of grow, and right before they split, and then they just get larger and larger. And that's how, like we talked about, you can get 4 generations in less than 20 minutes, because look at how fast something like that is happening. Within conjugation, this is where cells are actually exchanging genetic information. And it's not typically like one gives and the other gives and they both receive each other's. It's typically like one is giving information to another bacteria. But E. coli can also exchange information this way. And you'll see this picture over here is an example of that happening. It's typically the pili are what's involved, and you'll see them attaching to the other cells there and swapping information in that way. So we've talked about how they reproduce, we've talked about their structure. Let's talk about just in general how they survive, right? Because they don't just reproduce, they have to actually live. So there's 2 types of troughs within the bacteria. There's a photoautotrough and a chemoautotrough. Photoautotrough, just as you would kind of assume, they do photosynthesis. This is how they release oxygen into the environment. And super, super common is cyanobacteria, or also blue-green bacteria, which were formerly called blue-green algae, but these are responsible for tons of oxygen release into the planet and the atmosphere. Chemoautotrophs, on the other hand, they make their own organic food chemically, though, from organic chemicals. So something important here that we've talked about already is nitrogen-fixing bacteria. These are chemoautotrophs. They take nitrogen and they convert it, and that's how they also make their food. And then Archaea bacteria also do this process. Within that, we also have the difference between aerobes and anaerobes. And this should be kind of a review from you back to cell respiration. So we have aerobes, or aerobic respiration, uses oxygen. And then we have anaerobic or anaerobes, and these do not use oxygen. They're going to use that fermentation. So if you remember back to cell respiration, we go through glycolysis, and then if there's not oxygen present, the cells go through fermentation to produce ATP. That's how this process works as well with an anaerobic bacteria. Bacteria are also pretty cool evolutionarily. So obviously their fast reproduction rate is super helpful. We're going to start actually with mutations first. So if there's a mutation and if it's beneficial, that bacteria reproduces so quickly that before you know it, that mutation is in the population and it's helping these other bacteria survive. So these survival things, such as antibiotic resistant bacteria, which we'll talk about more in a minute, are easily passed on, and then those population numbers increase rapidly because of the way bacteria reproduce and how quickly they do. Endospores are another super helpful option for bacteria. So these are actually kind of scary when you think about it, because they're these dormant cells that form within a bacteria. And if you look over here in this picture, you see these like little key-shaped structures are actually endospores on the end of a bacteria cell. And then the oval, like the vegetative cell here, that oval, that's just a normal bacteria cell. It does not have an endospore in it. And the thing with these endospores, it's great for the bacteria. It's a great evolutionary advantage, but it's terrible for us because these cells can last millions of years until the environment is preferable enough for them to survive. And then these endospores come out of their dormancy and then they start to create problems or sickness again. And so really, really common Endospore bacteria are the ones that cause anthrax, botulism, and tetanus. These are all very bad. They make us very sick, but they can also be super resistant and can be around for millions of years before they cause problems again. So we've talked about all the bad bacteria, but honestly, bacteria gets a really bad reputation from us because there's more helpful bacteria than harmful bacteria, and we don't really think about how much we need them on a daily basis. First of all, decomposers, right? That food chain, the food webs, we need bacteria to break down dead plants and animals to recycle those nutrients back into the soil, into the atmosphere, so those cycles can continue. But also, normal flora, you have hundreds of millions of bacteria in your body every day, and they help you. They're good bacteria. They help you with digestion, they help you make vitamins within your body, and they also kind of give your immune system a boost. And then medicine. So of course we take antibiotics when we have a bacterial infection, but these antibiotics were originally, some of them were made from soil bacteria. So if you've ever taken tetracycline, this is actually originally made from a soil bacteria. And then lastly, a food source. If you like yogurt, cheese, buttermilk, even fermented foods, sauerkraut, sourdough bread, chocolate, kimchi, these all have bacteria in them, and we need them to create these foods. But of course, we have to talk about the harmful ones with the helpful. So like I said, only really a small percent of bacteria are actually causing disease, but when they do, it's bad. And they can harm their host, like us or animals, in many different ways. But the two main kind of categories are, one, they multiply really quickly, so they spread quickly and invade the body. And two, they can secrete this toxin that actually harms the local tissues around. And then food poisoning, if you've ever had that wonderful thing, that's also the result of bacteria, usually contamination, not washing hands, not washing utensils, raw meats, things like that. And then we also have things like strep throat, meningitis, abscesses, and some STDs that are all caused by bacteria. So lastly, let's talk about antibiotic resistance. And this is really important, and it's something that's becoming more and more mainstream as we go because we take antibiotics too much. We take them for things we shouldn't, right? We take them for viral infections. You don't feel good. You go to the doctor. You're like, I want antibiotics. I don't feel good. I need something, even though it's not really going to do anything for you. Or you do actually have a bacterial infection and you take antibiotics, but you're like, I'm feeling good 2 days later. I don't need to take 10 days of antibiotics. This is ridiculous. And so you stop. Well, guess what? You only killed, if you look at this infographic over here, that was before, you didn't kill these guys. So now guess what? They're going to reproduce and be in your body. So the next time you get sick, it's going to be even worse, and you're going to need a stronger antibiotic if a stronger one even exists, right? So the issue here is that traits that these ones had to survive that first treatment of antibiotics, they're going to pass it down. So they reproduce. Well, now you have all these antibiotic resistant bacteria in your body to where you can't treat them. So things like MRSA, if you've heard of that, that's a really hard staph infection that's resistant to antibiotics. Pneumonia, some gonorrhea, tuberculosis, all of these things are becoming more and more antibiotic resistant, which is doing more harm than good because it's also making things like organ transplants, chemo for people with cancer, and even C-sections much more risky because there's not necessarily a treatment if someone gets a bacterial infection. So antibiotic resistance is a super important concept that you understand, but you also need to understand when you take antibiotics, which is when you have bacterial infections only, and to always follow the recommended dosage. Don't just stop because you think you feel better or anything like that. All right. And with that being said, that is all for your notes.